First, I want to thank 100% Natural, or 100% Natural, Nutrisan, and Caneca for supporting this work. This work has been taking for 3 years, following 2 seasons in the Athletic de Bilbao team. This is a professional, and during this period has been playing in 3 competitions. One is National League, and another national is the King Cup and European Cup. So the objective was to see if the supplementation of coenzyme Q based on Ubiquinol is able to improve some aspects of the activity of the players. You have to consider that we don't know who is taking coenzyme Q. This is a volunteer, but we know some of them were taking at least for 2 periods of the years. So independent of who is taking or not ubiquinol, we are analyzing the levels of ubiquinol in each player, and we work on the idea of correlation between the improvement we see in the player related to the contents of plasma coenzyme Q. Okay? The plasma coenzyme Q you are going to see in the presentation is the total coenzyme Q, but we provide to the players ubiquinol. Okay? In terms of to understand what is coenzyme Q, here you see the 3 forms of coenzyme Q. This is the form that is more stable, is the oxidized form. It's called ubiquinol, it's yellow, and so if you buy a cream with a coenzyme Q, if it's not yellow, it's probably not present or very low. So this is reduced by 2 pathways. One pathway is, for example, in mitochondria, in the complex 1, and also in a very important enzyme for surviving and longevity, cytochrome B5 reductase. These 2 enzymes, and some others, reduce 1 electron first, then induce the production of semiquinone, is radical unstable compounds that immediately is reduced by another electron to produce the final form is ubiquinol. But for example, NQO1 is an enzyme, antioxidant enzyme in all the cells, provide 2 electrons at once. So never produce semiquinone, is come back and forth with this enzyme from ubiquinol to ubiquinol directly. This is important because this pathway or these reactions protect the cells against the radical and the electron, the localized electron that normally is present there. And if you don't have the right substrate to capture or provide the electrons, then can be damaged. But in a health cell, it's not happening, but can happen. Okay. Coenzyme Q is a lipid, so never is a solute in water except in some specific production, like a heating, like the milk, then it's cycled and then can be soluble in water or some other compounds. But normally it's a lipid with a change, isoprenoid change, that is inserted in the lipids, in the membranes, and stabilize the molecule. So the molecule is stable because it's there. And then they have a benzoquinone ring that is interesting because this connection can be moved back and forth. So when this is reduced, it comes out of the membrane. When the oxidized, come in. So this is the way to connect with other enzymes. This is the reduced, it is ubiquinol, and this is the list of potential properties that affect important pathways to maintain the life in the cells and also maintain longevity in aging process. The major and the first discovery was respiration, inserting the electron change between the complex 1 and 2 to the complex 3. So we cannot respire without coenzyme Q. So only some bacteria and yeast can survive without coenzyme Q, but during fermentation. The antioxidant activity is provided anywhere. Independent, you can move electrons back and forth. You are controlling the electrons and not releasing electrons from some reduced compound, and they can then stabilize, and also coenzyme Q can stabilize by themselves. The major antioxidant properties are in the plasma membrane, in the membrane that surrounds the cells, but also in the lipoproteins. So the major place for antioxidant protection by coenzyme Q. The uncoupled proteins, this is in Spanish, but the uncoupled proteins is proteins that are present in different tissues in the electron change in the mitochondria, and when there is a excess of production of transport of electrons, uncoupled proteins move electrons back from there to prevent excess of damage. So uncoupled protein is responsible, for example, to maintain temperature in the bear during hibernation. And also the babies, when they are born, in the first weeks of their life, they cannot heat the muscle to produce heating, but they depend on the small amount of brown tissue that using these proteins maintain the temperature. Okay, this is very important in some times of the life of some animals. The transition pore of mitochondria is when the inner membrane and the outer membrane fuse, and then they can modulate the activity of mitochondria and normally tends to be recycled. It's eliminated to produce new ones. And this activation normally is happening when there is a toxic effect or some kind of damage or infection that the cells are not very healthy, and then you have to remove the mitochondria to generate new mitochondria that provide energy. The synthesis of pyrimidine nucleotides is important because mitochondria contains DNA. To make DNA needs pyrimidine nucleotides. So coenzyme Q is inserted and its obligation pass to produce pyrimidine nucleotides. And the beta oxidation fatty acids, for example, is a muscle is using fatty acids for most of the movement. If you have 100-meter say contest, then you don't need probably very much fatty acids because it's an anaerobic movement. It's done by muscle or reserve of energy in the muscle. But when you do a marathon, it's a long way, then you have to use those bioenergetic compounds that require the most energy production, and this is fatty acid. So fatty acids pathway, it depends strictly of coenzyme Q. This is an example or an idea that we can send you the scheme. How this is the electron chain. The electron chain is formed by 4 complexes that produce a movement of electrons through the coenzyme Q, reducing coenzyme Q that then send electrons to the complex 3, and the 3 through the cytochrome C to the complex 4, and the complex 4 is using the oxygen you get when you are breathing. Okay, the CO2 is produced before that in the glycolysis. Here, you produce water. So all the oxygen you get from breathing in the complex 4 using the electron from coenzyme Q go to produce water. But complex 1, 3, and 4, and some people say that coenzyme Q itself, but not the complex 2, produced by the movement of electron in this way, produce a work that moving protons from here to the interspace. And these protons accumulate here. Think if you have here 5 protons, and you have here 5 protons, because in all the membranes are equilibrated, and you, using this strategy, move protons to here, then you can get here 9 and 1. But naturally, in the stoichiometry, try to move back protons to the original space to equilibrate again. But that happens through complex 5. Complex 5 is no electron change. It's a component of the production of ATP. So the protons come here, are accumulated here, and they find a hole here. And this hole moves protons back to their right place, and in the meantime, you can move moving there to produce the fusion of ADP and protons, and produce and phosphate, and produce ATP. But when you have an excess, by some defect, an excess of protons, or for example, very cold temperature in the bear, then there's proteins that are uncoupled proteins that make a hole bigger with any difficulty, so protons try to go through this way before doing this way. Or it's a preferred way to come back. And this pathway induces no ATP production, induces heat. And that's why the electron change produce temperature, higher temperature for this animal or the baby. There is another enzyme that this is the BDAX part of the transition pore that normally depends on coenzyme Q because require the structure to move back and forth some ions and some other. But it's a part of the mechanism of mitochondria, but no ATP production. In the plasma membrane that you see here, there is an important component, antioxidant component. This is vitamin E. Vitamin E can be in the reduced and oxidized form, but there is no any enzyme able to reduce vitamin E. So if you don't reduce vitamin E, it's degraded, it disappears. That's why we need to get vitamin E every day. Okay? But we don't get enough vitamin E that we need. We get less than we need. Why? Because part of the vitamin E is reduced by coenzyme Q, Ubiquinol, in the plasma membrane that is reduced by reductase like cytochrome B5 reductase and Q1. Okay, to protect the membranes, coenzyme Q is a factor because it can be recycled by the enzymes, and then secondary recycling components like vitamin B, C. And this prevents the lipid peroxidation of the membranes. There are some examples of these reductase are in the electron change in mitochondria. Outside mitochondria are these major enzymes, that is cytochrome B5 reductase NQ1. This NQ1 is immediately activated when you are in stress, oxidative stress or other. And this is cytoplasmic NADHQ reductase is a pool of enzymes that probably most of them are these two. But there are 6 in the library, in the literature, and we have to put here. And then there is another enzyme that use coenzyme Q as a substrate, not very much specific, but they are in the cytosol, and all are antioxidant based on glutathione. Another aspect that is outside the cells are the lipoproteins. This is a typical scheme that you have a lipoprotein LDL or HDL, and when you induce the oxidation, for example, you are smoking, having a glass of rum or wine, okay, so in this moment you are inducing an stress, oxidative stress in the blood. And this captured by the proteins and vitamin E, for example, here, and very low coenzyme Q, and immediately is oxidized. The white one are oxidized. But if you increase the amount of coenzyme Q inside the lipoproteins, after the radical damage, for example, smoking, then it's more protective. Why? Because coenzyme Q, again, can be reduced by enzymes. And 100, let's say 100, but 95 to 98% of the coenzyme Q in the plasma is usually reduced as ubiquinol. So when you come the stress, if you add more coenzyme Q, then you can reduce vitamin E there. Lipoproteins contain a lot of vitamin E, but also itself can prevent damage. So this is important by supplementation of coenzyme Q in the terms of ubiquinol is very important to prevent the oxidation of lipoprotein, particularly in hyperlipoproteinemia. For your understanding, this table was published in 2011 of the total coenzyme Q contents in different tissues of the human. The highest is the heart. Muscle is very important, is not the biggest one, and kidney. That's why when you have genetic defects that produce a very low concentration of coenzyme Q, most of these tissues are affected. So by some reason, I don't know why yet, I expect to understand soon, that heart have a protection by itself and induce overexpression of some enzyme to produce more coenzyme Q. But liver, kidney, and muscle and brain are the most sensitive tissues when a gene defect induce a very low content of coenzyme Q. And this is also important because I think the area of ubiquinol supplementation is going to be aging. Aging is huge. Many laboratories are now dedicated to aging analysis to prevent aging. But you see how coenzyme Q, internal coenzyme, endogenous coenzyme Q, is coming down during aging in some tissues more than others. But you see, kidney is very important because kidney filtration in kidney depends on the treatment of ATP, high content ATP. And that's why the kidney get old like you. And the transplant of kidney is cannot be done for a guy like me because the kidney age is my age. So it's different of liver or heart that maintains a kind of healthy condition. So I think in terms of maintain the activity of the people during aging, we need to do exercise that provides some preventive. This one prevent smoking, doing walking, but particularly after 50 or 60 years, it's important to consider supplementation of coenzyme Q, but always after an analysis of the real contents in each one, because this is the average, and we need to know each personal content of coenzyme Q. And this is the second part that is going to be presented by Guillermo. He has been working hard with all the data coming from the players, and this is the specific part that he is starting now.
Okay, I'm going to show you now the results that we have been working with the plasma from professional players in the Athletic de Bilbao team during 2 different seasons. First, we worked with the season 2014-15, and we determined the several parameters that can be related with the coenzyme Q or ubiquinol level in plasma, because as Placido told, the 95-98% of coenzyme Q in plasma is in the reduced form, it's ubiquinol. And here you can see that during this season, in the preterm, it was used at the beginning when the players returned to the team to start the pre-season. And then used at the first matches of the season, could be the second or the third matches, because they start playing at the end of August. And then just at the mid-season, when the stress during the competition is very high, because they play 3 different competitions, and then in more or less in those months is when all the finals or the pressure to get points during the league is higher, so they are very stressed. So we can see here that the levels of consanguine Q increase just during this part of the season. There was the stronger one. We don't know why they have a very high level during the pre-season, but this is not important for a study, because we can compare easily just in the beginning with the mid-season. Also, the levels of consanguine Q related to the cholesterol forms in the plasma are very important, because most of the 100% of the ubiquinol is linked to the cholesterol. No other proteins or carrier are involved in this. So we can see also that are increasing the levels of consanguine Q related to the cholesterol. And also we can find that the levels of stress or oxidative stress is very low. The T-bars is how we measure the oxidation of lipids in plasma. It's very low, it's not significant, it's no changes at all. But the most interesting thing was that the levels of creatine kinase, that is a marker of muscle damage in the players, increased just when the season was at the middle, so just when the pressure of the season was very high. We also we made some correlation between consanguine Q and the levels of creatine kinase, and find that in September there was no pressure, they just start playing. There was no relationship, but there was a very low relationship. But when when the stress is high, the levels of creatine kinase show a inverse relationship with consanguine Q. This means that if you have lower consanguine Q in plasma, you have higher muscle damage during when the season needs more than for you. We also have some results from AXPE laboratories that make the routine determination in those players, and also found that in those parameters there was nothing significant in this season, but we found also that this marker, that is a transaminase. Transaminase can be in the plasma not only because you have liver damage, also because you have muscle damage, and was more or less similar than we have found with creatine kinase. That was higher during the season that was at the middle. Well, we checked also in the next season, that was the 2015-2016. And during this season, we have to say that the numbers of players that took Ubiquinol was higher. During this, the first season, the number was low, but now the number was higher. I think the double of players take. Not all the players took consanguine Q, but most of them in this case took the play. And then we can find also the same. And here you can see, this is we took 2 points during the mid-season. And you can see here that the level of consanguine Q in plasma was very higher than in first season, because most of them took consanguine Q. Also increased the level of here. We didn't check the oxidative damage because it was not significant in the first season. And also we can find that the levels of creatine kinase, in this case, didn't increase during the mid-season. So they don't increase, probably because people have more consanguine Q. We also have data from AXPE laboratories and found more or less the same. There was an increase also in this transaminase, but was not so high. Also, we have here the levels of cortisol, that is a marker of stress, and in this case was lower than during the first part of the season. That was very interesting because It's in that the stress is lower, and also other markers that can be can indicate some damage in liver or damage in kidney. They were not significant. There was no changes, so it means that the coenzyme Q didn't make anything in the normal metabolism of the players. Here is the comparison between both seasons. Here we can see the levels of coenzyme Q, that I have to repeat, is ubiquinol in plasma. In the first season in white, in red in the second season. You can see here the increase during the second season. Here is the comparison just at the middle of the season, that is very significant. And here the relationship with the creatine kinase that indicates the muscle damage. And as you can see here, during the first season, when the coenzyme Q levels were lower, the amount of creatine kinase was higher, but when we increased the levels of coenzyme Q, the amount of creatine kinase was very low, and then no change along the whole season in the second part. This is the comparison between both seasons. And here, very interestingly, we correlate the levels of coenzyme Q in the players with the levels of creatine kinase during both seasons. And here you can see here clearly that it's an inverse relationship between the levels of coenzyme Q in plasma and the levels of creatine kinase in plasma during the mid-season, when the stress is very high. This means that in some way, ubiquinol in plasma is protecting muscle from damage when you are requiring more activity in the players. Here is the relationship between the levels of coenzyme Q. We determined more or less the middle of the level, determined that people that have less than 800 nanomole per liter in coenzyme Q can suffer more damage in the muscle, and people that have more than 800 nanomole per liter in plasma are more protected. Why we decide that? Because it's more or less the level that determines that higher levels of coenzyme Q in normal population and lower levels of coenzyme Q in normal population. Also, in the case of cortisol, we can see more or less the same. In the second season, the levels of cortisol were lower during the mid-season, and it was very significant. And also, there's a correlation between the levels of coenzyme Q and cortisol, but it's not significant, but it's a trend that indicates that coenzyme Q in some way protecting. Also, if you have lower levels of coenzyme Q, you are less protected. You have more coenzyme Q in plasma, you are more protected. And another part of the study was to determine, okay, is ubiquinol increasing the capacity of the players, or is in some way related with the capacity of the players during the team? And here we analyze the activity of the players during the league, taking all the parameters that are important during the league for a team, football. That is the amount of meters that the people are running or during the play, and also the mean speed during the play. And here you can see the population. It's very strange because there's two people that run very low and the speed is very low, but they are the goalkeepers. So it's normal, the goalkeeper doesn't run. So there's a correlation, but in this case also there's a correlation, positive correlation. It means that if you have higher levels of coenzyme Q in plasma, you can run more, and you can run faster, or you can move faster. If we remove the goalkeeper, because it's normal to see that, in the field players, you can see that this correlation is not significant. Could be because the number of people in the play is lower, is 20, between 20 and 22 people in the whole league. But this has a positive correlation between the amount of coenzyme Q, and if we determine the levels of coenzyme Q and the capacity to run, If you have less than 800, you run less, and you run lower and less speed than if you have more constant Q. And this is near significant, but not at all, but it seems that there's a trend, a positive trend. But there's some differences between the matches. A difference between the first part of the match and the second part of the match. And it's very interesting because in the first time, the differences are very, very, very significant. So it means that the people that have higher constant Q during the first part of the time, that in all the teams, they run more and they run faster, there's a near significant positive correlation, but in the second time, the differences of more or less similar but people that have more constant Q in plasma have a bit more capacity to run. So it seems that, okay, there's no clear effect of constant Q in the second part, but in the whole match, there's a positive correlation. And this is more or less all the parts of the study that we have made. And the conclusion was that the supplementation of Ubiquinol is safe, doesn't produce any damage in liver, kidney, and they are very happy. There are inverse correlation between the levels of constant Q and the levels of creatine kinase during the strongest part of the competition. It means that there's more protection with constant Q. Also with cortisol. And there's a direct relationship between the levels of constant Q and the aerobic capacity of the players, determined by the mean velocity speed and the distance covered by a player during the match. In general, we have that a higher constant Q in plasma, that is Ubiquinol in plasma, we have higher physical capacity during the matches, less muscle damage. This is very, very important, and it means that the Ubiquinol can act with an efficient factor to the professional players in football. And that's all. Thank you very much. This is our laboratory, and we have to send the whole group, the expert laboratories, people in 100% Natural and Caneca, and the people in Athletic de Bilbao. Thank you very much.
First of all, on behalf of Nutrisan, I would like to thank you, Professor Navas and Dr. Luch, also for the interesting presentation that you gave earlier. Now, although the coenzyme Q10 is not a new molecule, I have the feeling at this moment that we don't have an idea, or we don't know the full potential of the molecule at this moment. Now you, Professor Navas, as president of the International Coenzyme Q10 Association, what is your opinion about that?
Okay, thanks for having me here with you. And a pleasure to explain what I know or what we want to know now. But coenzyme Q is an old molecule. The problem to understand coenzyme Q is new because we cannot live without coenzyme Q. So it's impossible to find an animal model or a people without coenzyme Q. But recently, from the 90s, came some patients with very low coenzyme Q and opened the possibility to understand what it's doing when coenzyme Q is provided to the people to rise the level. But still many questions are open. For example, when you take coenzyme Q by supplementation, many people don't see a rising level after some hours in the blood because they have to reach the tissues. Mm-hmm. So we had to understand how the external coenzyme Q is providing protection and activity in the new tissue. So the new samples or the new models of animal models like mice that have been developed at this moment, there are 5 only in the science, that is used to understand how reach coenzyme Q to the tissues. And this is important because in aging process, coenzyme Q is decreased, and we need to know how we can improve some activities like immune system or antioxidant protection by providing coenzyme Q.
Okay.
Because many of this information is still incomplete.
Yes, and unknown.
Incomplete, I say incomplete. Because there are some information from patients that were dying very, very soon, and the tissue were analyzed, but not very much. Okay. Or, for example, the cardiac surgery, the piece of heart that they are removing sometimes, they are analyzed. Or the transplantations. But few tissue, because brain, you are not transplanting brain, you are transplanting kidney, you are transplanting heart, and then the analysis of this tissue demonstrates a decrease and the relationship or the negative correlation with activity of the low level of coenzyme Q.
Yeah.
That's, I think supplementations is important, is going to be important in this kind of disease associated with aging.
Now, if you're talking about supplementation, and now recently several studies has been done with coenzyme Q10 on fertility, on chronic fatigue syndrome in sports. Now, in every single study, the reduced form of coenzyme Q10 is the ubiquinol is being used, and in none of them the ubiquinol, the oxidized form. Now, can you explain a little bit more about the difference between the both and why you have chosen to use ubiquinol for the study at Athletic de Bilbao?
Yeah, using what you do supplementation with ubiquinol, if you do analysis very fast, then you see that the plasma of individuals rise in a few hours. But not only because you are using ubiquinol, because you are using a support, a vehicle of the ubiquinol that also take place in the velocity that reach plasma through the intestine. So it's not only ubiquinol that is immediately incorporated in the activity because most of the plasma coenzyme Q is ubiquinol, but also by the way that is prepared to supplement the people. But I think ubiquinol is also analyzed in animal models. In animal models, we don't use the vehicle, we use the food, the same food. So we don't have an intermediate to facilitate transportation. And we have demonstrated, it's going to be published, but also some other people, that if you provide supplementation in the food to the mother, for example, it's reaching the puppies before they are taking any food, only the milk. So this is important that coenzyme Q can be provided through the mother to prevent damage that are happening before born. Okay. This is very important. This is very important because many of the disease, when you detect the disease or you make the diagnosis of the disease of the children are just born, but the damage accumulated during development, during pregnancy, you are not protecting them. We need to treat the mother. This is the idea of the future. And this is important of the supplementation and also aging. But I talked to you about aging. I think ubiquinol is faster when you provide through the food. Yeah. So you don't have the absorption.
Ubiquinol is much better than ubiquinol, and that's the reason why you want to supplement with ubiquinol.
Exactly. So you have to distinguish between supplementation to humans that are in a pill with some oil or some other vehicle, compared with the animal models where you don't use this intermediate of transport, but just right on the food. And in this case, ubiquinol is very efficient compared with ubiquinol and reach immediately, for example, in the pregnant animals, immediately reach the tissue of the baby. Yeah. So this is important. We have to think about ubiquinol incorporation in models and then translate this to human, because they are different.
Yes, of course. Okay, very interesting. Now, the outcome of the study done at Atletico de Bilbao shows a big potential of Ubiquinol in football.
Yeah.
What about other sports?
The big problem of the studies on, you can see in the literature, is the people trying to demonstrate the importance of coenzyme Q. Many work on Ubiquinol, some other Ubiquinol, but all of them are very short treatment. And very short treatment means that you have to adapt the tissues, you have to adapt the body, and this requires time. If you provide Ubiquinol today to a guy, and then to the analysis in plasma, you can see huge amount of coenzyme Q. But you don't know how much reach the tissue and how much are using by these tissues to improve the activity. So I have seen some presentation of short-term treatment, like weeks, and they don't see anything except antioxidant protection in blood. The work we have presented today, we demonstrate protection in the tissue, in muscle, and also in the production of transaminases, and also in the activity of the players, because we taking 3 years.
Now, the study is done with the football players of Atletico.
Professional
football players. Now, can we extend the outcome also to other sports? For instance, tennis players or marathon runners. I think it's practically the same process. So can we make the conclusion that we can also apply the same results for other sports?
Yes, I have seen data that are confidential, but I can say, don't say who, but some positive effects in rugby, European rugby, and semi-professional team. I have seen some benefits in running, but I insist, it's not taking one day, one pill, and then everything is fine. You have to maintain the level, and then each tissue, particularly muscle, adaptate to that and improve mitochondria and improve the antioxidant protection, not only the blood.
Okay, so we learn from it that you don't take it for a short time. It's better to use it for a longer time.
For a long time, to maintain the level. Any tissue, any exercise that require aerobic activity will be improved by a treatment with Ubiquinol.
Okay, that applies to many sports.
Most of the sport.
Yeah. Okay.
Most of the sport. Tennis, for example, could be important.
Yeah.
A marathon could be important.
Yeah.
And for example, the American football is little different because they run very fast and they stop and relax and start again. But the rugby or the soccer, it takes 45 minutes back and forth running with a few stops.
Yeah.
And you have seen the data of the goalkeeper.
Yeah. Yeah.
You see, it's totally different. They are out of the effect.
Yeah. So besides the protection of the ubiquinol of coenzyme Q10 on the muscle damage during exertion, it also has other benefits. And so like you presented in the study in the presentation earlier, what specific things are also beneficial when you take the ubiquinol?
Well, we have seen the capacity of the player to maintain the activity with more intensity, longer time. For example, one thing that you see anytime you watch a football game, you see this guy ran 8 kilometers, 9 kilometers, and we have demonstrated that they can run more when you have enough amount of coenzyme Q in the blood. This is probably not the best correlation because we need to know how much they have in the muscle. But if we accept that if you maintain the level of mostly is ubiquinol reduced in the plasma, you are considering that they are providing any tissue, including the muscle, because in mice, plasma ubiquinol rich muscle, so we can consider it in human also. So this, if you maintain enough ubiquinol long time, they show more capacity, running more speed in the second time of the game and longer time at the end of the game. Okay. Because the data we have presented is the people that have been playing all the game, because if you have 10 minutes, we have removed for that.
Yeah, okay, interesting. Now, can we consider coenzyme Q10 in general safe and doping-free?
Totally, totally. This is a compound that is in our body. We produce in our body. Cannot be a doping. Because that is demonstrated in human cells when you provide ubiquinol. If you are healthy, you reach or increase no longer than 30% or so. That is to the maximum capacity of the body. And the rest of the body synthesis, it comes down. But you use very much coenzyme Q, then sometimes the body cannot synthesize with enough speed to complement, so it requires some time. So supplementation can reach the total, but is not a doping because you have your own source of coenzyme Q.
Can you take too much coenzyme Q10?
No, it's eliminated.
Okay, so you have a maximum absorption rate. And
your capacity of absorption, yeah.
The excess will be removed.
Yeah, coenzyme Q, there are few cases, but there are some cases of people that suffer the neuropathy, myopathy, and some of them dying very soon. I'm dying with 15, 16 years old. We published 2 cases in this condition, that they have excess of coenzyme Q. Because there is no mechanism to block the right content by some protein defects or whatever, and they die. They have the same phenotype, pathological phenotype, than people that show very low coenzyme Q by the genetic defect. So excess is bad, low is bad, so this very much equilibrate, adaptate to the capacity of the rest of the components. So it's not a doping, it's adapted. If you have an excess, it's eliminated by the kidney, by urine, and you get what you need to complete your maximum capacity.
Yeah. So what would be the recommended daily dose when you do sports? It depends a little bit on the intention of the sport, I assume. What would be the normal recommended dose when you take in sports?
It should depend on each one. This is the first answer, because I want to say that some of the people that you have seen here in the study, we know that some of them didn't take ubiquinol at the high level, because their own production, okay, is higher than that average. For our patients, we recommend 30 milligrams per kilogram a day. That they need to maintain some activity. So I would recommend a similar amount. The people that are taking 300 milligrams a day, they are doing nice activity, not in professional, because we provide them 300, but this is the only study in long term. But many people that are non-professional, they are taking 300, normally 150 and 150, but never to have the 300 together at once, in 2 or 3 times. And they are improving their capacity. For example, it's a kind that we didn't get yet the data, is the painful. The pain you have here, that people with simvastatin or statins that have pain and require coenzyme Q. But painful is also a good marker to know how much do you need. Yeah. If you consider 30 milligrams per kilogram a day, and you disappear, you are paying, you are doing exercise, that's fantastic, because this is a marker that you can establish by yourself.
So it's a little bit subjective from the sports people.
For the sport people.
The sports people. Okay.
For the sedentary people is another story, but for the people with high physical activity.
Yeah. Okay. Now, can we conclude that the coenzyme Q10, Ubiquinol in general, will have a prominent place in sports in the future? What do you think?
I think that it's going to be important when you have some age to run, to do activity. Maybe with 18 years old, maybe not so important if you are not very much intensive, but most of the people are from 30 to 50 doing sport, or in age people, that they had to maintain a physical activity to maintain the muscle. It's important. Yeah. It's very much important. So I say for the range from 20 years old until 60, 70, this is going to be a future supplementation of coenzyme Q after the analysis of the level each one has.
Okay. Well, thank you very much, Professor Navas, for the interview, and thank you again for the presentation. I also want to thank our distributor and partner in Spain, 100% Natural, to make it possible to be here and to have this presentation and this interview done. Okay, thank you.
Okay, thank you very much. I would like to thank you to give this opportunity, and if you want to any other questions sent to me, I can answer you immediately.
Okay, perfect. Okay. Thank you very much.
Thank you.